Okay, and this lecture we've already covered, for chapter 10, we've already covered the structure of genetic material, we've already covered DNA replication, and we'll cover this stuff later. So basically this whole lecture is going to be about the flow of genetic information from DNA to RNA to protein. That's the big thing here. Alright, so flow of genetic information from DNA to RNA to protein. Okay. Alright, we'll say the essential question is, how are proteins made? We'll just say using the genetic code. So a lot of this is going to be like DNA replication type business, but it gets a little more complicated. We're going to have to add on to it. So think about what you did in your DNA replication stuff. Maybe think about, you may even turn to that page occasionally to sort of look at what's going on, but we start kind of on those basics and move from there. So DNA specifies traits by dictating protein synthesis. So the long way to say this is that kind of proteins are the links between the genotype and the phenotype. So the DNA, the actual DNA, gives us the genotype, this sort of genetic code. But proteins actually will help us get to the phenotype. So DNA only specifies for protein, but those proteins are what help us lead to our physical appearance of things, or our phenotype. So lots of scribbling on the screen. Let me know if that doesn't make sense to you, but we'll try to review that. So the molecular chain of command is from DNA in the nucleus to RNA, and then RNA in the cytoplasm to the protein. So let's look at a diagram for that after we give it some terms. So transcription is the direction, or is the synthesis of RNA under the direction of DNA. So if you think about the word transcription, so it comes from the word script, or kind of transcribe. So trans means change, and so we're going to change the writing, because script is to write. So you can also think transcribe. So we're just changing the writing. We're still under the language of nucleic acids at this point. Translation, however, think about translation. You think you have to translate from English to Spanish. Well, we're going to translate to the language of proteins from the language of nucleic acids and RNA. Okay? So that's to keep those straight. Here's a good diagram. Let's do this completed diagram. It'll be a couple of slides here. If you draw it out for me, you'll get that extra credit for that test, which we'll need 2 parts. So the Easter egg this time is to draw this completed diagram. So this is what we call the central dogma. We start with DNA. From that, we're going to build an RNA strand in the nucleus, and this is called transcription. We're rewriting it from one nucleic acid to the next. Because DNA can't leave the nucleus, we will allow that RNA to leave the nucleus through pores, and then we can use it to translate or change the language into a protein. So this would be the bit that I want you to draw. You might draw it for yourself, but you also would do that for extra credit from me. Figure that one out. So DNA to RNA to protein, transcription, translation. And we'll go into each of those processes individually. So genes provide the instructions for making specific proteins. The initial one gene, one enzyme hypothesis was based on studies of inherited metabolic diseases, but the one gene, one enzyme hypothesis was expanded to include all proteins. So really it's kind of like one gene, one protein. One gene will lead us to one protein. So genes lead to proteins. That's about all we're getting from that. I think that's just proteins being synthesized under a petri dish condition. I forgot what that one was about. Doesn't matter. Most recently, one gene, one polypeptide. And remember, a polypeptide is sort of like a pre-protein. So we can start with that. It's just not folded to the right shape. Was recognized that some proteins are composed of multiple polypeptides. So even this description is not entirely accurate, and that the RNA transcribed from some genes is not translated but nonetheless is important, has important functions. So we could sort of give it the general one gene, one protein kind of thing. Just know that it's not 100%. I mean, that's most of science anyway. In addition, many eukaryotic genes code for a set of polypeptides rather than just one by a process called alternative splicing. I'm going to show you splicing in a bit, but let's not worry too much about alternative splicing, although it's neato mosquito. So the sequence of nucleotides in DNA provides the codes for constructing a protein. Protein construction requires a conversion of nucleotide sequence to an amino acid sequence. Transcription will help us rewrite the DNA code into RNA using the same nucleotide language. So that's essentially the stuff I gave you already anyway. So what's up on the first one is a good description for translation, if you need another way to describe it. And this is indeed transcription. So this should go first and then this one second. So the flow of information from gene to protein is based on a triplet code. The genetic instruction for the amino acid sequence of a polypeptide chain is written in DNA and RNA as a series of non-overlapping 3 base pair words called codons. C-O-D-O-N-S. So codons, there the translation involves switching from the nucleotide language to the amino acid language. Each amino acid is specified by a codon. 64 codons are possible because we've got 4 bases and we've got 3 possible positions for them. They can repeat. Some amino acids have more than 1 possible codon. So here's to look at kind of the ideas that we've got multiple genes along the kind of DNA strand. And in that one section, we'll have DNA, and we have A, C, whatever. We've got all these, and we've got our A's and T's and G's and stuff. It will get transcribed or rewritten to the language of RNA, which still sort of looks the same. Same basic stuff, still matching up together here. But remember, RNAs have U in it. If I could choose, that have RNA because it has U in it, right? Translation goes from the RNA to the polypeptide, through a 3 base pair sequence, and that gives us our amino acid code. So we have to translate it to a whole language based on 3s. Okay? So the genetic code is the amino acid translation of each of the nucleotide triplets. Okay? Genetic code is the amino acid translation of each of the nucleotide triplets. 3 nucleotides specify 1 amino acid. 61 codons correspond to amino acid. AUG is always going to be our start, and there's 3 stops. Honestly, that's just as we kind of come across it, so don't worry about that too much. Fascinating stuff, though, that's at least worth noting. I'd say read it. So the genetic code is actually redundant. So more than 1 codon code for the same amino acids. This is a good thing to help us minimize mutations. So it's for minimizing mutations, by the way. A lot of things are in place to help us minimize mutations, because mutations are going to happen. We just need them to not have a very big effect. They are unambiguous, and that any codon for 1 amino acid does not code for any other amino acid. So 1 codon is 1 specific amino acid. And this stuff is nearly universal, and that the genetic code is shared by organisms from the simplest bacteria to the most complex plants and animals. Like, there are some exceptions, so much so that I don't even remember what they are. So there are exceptions, I just don't remember. Like, I remember hearing it go, like, wow, that's cool. But most things just follow this genetic code. Okay, so here's kind of what we're talking about, which is cool stuff in this one, is if you'll notice, like, CCG here only does for proline, but I have CCC, CCU, they all do for proline as well. This is a good thing. So we have a little diversity here, but there's so there's multiples there, but that one particular set is only going to code for one thing. Here's our AUG, which is always the start codon, and there are 3 stop codons, which tell us to stop a sequence. Okay, we're going to talk through this chart and how it works a little bit later on, so we'll practice. But here's our strand to be transcribed. It will start with a DNA strand, and we will have to practice these because they love this crap on the STAR test. True story. Okay, so we start with a strand to be transcribed, and then we do transcription, and so from this, we'll get them matching, like here's our matching thing, and we'll build AUG, AAG, UUUAG. Boom. We just rewrite it into an RNA, kind of eliminating the T's. Then we'll translate in sets of 3, AUG to methionine, AAG will be to, I think that's the lysine, UUU to phenylalanine, and then UAG will be a stop codon. So that's sort of our process. And we get these genetic mutation things that we're doing. So for some of you guys doing GMO stuff with GlowCats, that's part of where that's coming from, is that we're altering that genetic sequence to be able to alter the proteins that are produced. It's good stuff, which we'll come to that. Okay, now we're going to go through the sequences of each of them, which isn't too hard, but it can be a lot. So I would definitely suggest you take a couple-minute break on this one, and then come back and really follow along with me on what the important steps are. So now is definitely a good time to break, pause, look over the notes, write a summary, make sure you get what's going on to start with, because it's going to get more complicated and detailed from here. All right, so transcription of a gene. So we're in that first part. We're going DNA to RNA. That's what this whole DNA to RNA is, our transcription stuff. So transcription of a gene occurs in 3 steps. There is initiation, which just means we start it. Elongation means that it grows longer. And then termination means it stops. This is one of the more descriptive ways that we have a process. Yay to whoever designed the words for these. Good job, buddy. So initiation involves the attachment of RNA polymerase. So this is an enzyme that builds a polymer of now RNA instead of that DNA polymerase we saw before, and as a promoter to the start of the RNA synthesis. So there's a promoter, a little thing that just says, hey, RNA polymerase, start here. That's it. Elongation, the newly formed RNA strand grows. It gets longer. Termination. RNA polymerase reaches the terminator DNA and the polymerase molecule detaches from the newly made... Oh my god, we could shorten this. RNA polymerase reaches the end. Okay, and then it detaches. And then we have a new RNA strand. Okay, it's from the gene, if you really want to add that. But think of ways to shorten these, as we talked about. So initiation starts with, here's my promoter sequence. My RNA polymerase will attach onto that. It then will move in a 5' to 3' direction, actually building 5' to 3' as well. Ta-da! So it builds in that direction, 5' to 3' based off of a template, and it will elongate there. Then we get to termination, which it reaches the terminator sequence of the DNA, and it says, like, oh, we're done now. And then we're done. So that's that. And that's a quick process, but we will review it together. Okay, so hopefully you get the very basics of what's going on in each of those, because we'll review it, but we've got to move forward. Because Lord knows if I go over like 32 minutes, you guys panic on the length of the video, right? Just kidding. I do actually try to make them shorter. I'm trying. But there's a lot here. Can't cut all of it. Anyways, the thing that we just made is called the messenger RNA, the mRNA. It encodes for amino acid sequences. It conveys the genetic message from the DNA to the translation machinery of the cell. In prokaryotes, that's all in the same place because they don't have a nucleus, but in eukaryotes, the mRNA must exit the nucleus through the nuclear pores in the cytoplasm. That's the idea. So let's say, for example, because we're making a recipe here, and so I'm going to give you an example. On my mom's wall at the house, she has my grandma's old recipe for these chocolates called Millionaires. It's a big deal, these Millionaires, but I can't just take it off the wall. Like, my mom's house is the nucleus. I cannot take this thing off the wall. If I walked out of the house with my grandma's Millionaire recipe, like, I Actually, I just got a little scared on my insides thinking about what my mom would do to me. Like I wouldn't live to see the next day. Just kidding, maybe. But nonetheless, I'm not going to try it. It's going to sit on the wall, right? So if I want to get a copy of that, I need to rewrite it or transcribe it so that way I can take it maybe to the school or to some other place that actually has a kitchen and make them. But I can't take that recipe off the wall. She will kill me. I would be dead. All right. So the messenger RNA is like a picture that I'm taking with my phone, or I'm sitting down, because we'll use better language for this. So I'm like rewriting what is on the recipe. So rewriting the recipe gives me my own little mRNA strand. So that way, if I take my rewritten recipe and I go outside and it rains and it kills it, or I shred it up, doesn't matter. My mom's original millionaire's recipe from my grandma is still on the wall. Everybody's safe, especially me. All right. But, so that's the thing. I've got the messenger RNA. Like, I've got my rewriting of the initial copy. So this little copy that I took of my grandma's millionaire recipe, it has some introns or some interrupting sequences, and then exons, some coding regions. So the thing is with RNA is that you have to change it a little bit, and we've got to process it. It undergoes processing before it leaves the nucleus. That's what we call RNA splicing. It's going to remove introns or intervening sequences. You can think intervening, interrupting, any enter kind of word like that that's bad. And we're going to join and use exons called expressed sequences. So seriously, do not go with this. If you start changing exons and introns to anything else, it will not make sense in your brain. So they're intervening or interrupting, so we take them out and we express the exons. Okay. Then we actually put a cap and a tail on the nucleotides at the ends of the RNA to facilitate export, to make sure that we don't actually tear it apart once we leave, so we know that it's something important. It's like if I put my copy of my grandma's recipe in a sleeve and I bring it somewhere, so if I have it at school, it's going to protect it, and then we'll know, oh, well, that's something important, and I probably just shouldn't put that in the shredder. And it'll help us bind to the mRNA. So the cap and tail themselves aren't really translated into the protein. They're just there to help protect it. It's not what I use for the recipe. It's just going to make sure that I can protect my copy of the recipe. Okay, so here are the examples on this one. So the exons are the expressed regions, the darker pink, and I've got introns in the middle. The introns, the intervening sections, are taken out and we splice or glue together the coding sequences here. And then we can put this cap and tail on it, which I know it did kind of originally, but the cap and the tail are on there, and so it can leave the nucleus safely. Actually, introns to my grandma's recipe would be that, back in the day, they actually used to use wax in candies. And it's probably not a good idea. I probably want to take out the paraffin wax business. Me figure something else that you want for these candies, because I'm not sure wax is a good idea. We still got to edit recipes from back in the day. Okay, so on to the next piece of actually sort of making the food, if you will, making whatever from the recipe. So we're going to need some transfer RNA molecules. They're sort of like the tools to make food. Sometimes you're going to need little measuring spoons and whatever else. So these guys are there to help us function as an interpreter and help us convert that genetic message of RNA into the language of proteins. So they're there to help us build the recipe. So transfer RNA molecules perform this interpreter task by picking up the appropriate amino acid and then using a special triplet of bases called the anticodon to recognize the appropriate codons in the mRNA. So you could think of anti more as opposite. This is a good time to think of opposite. So they're the opposite codons. They're not against the codons. There's not a fight. They're not they're not at war. They're actually helping each other. It just means it's the opposite one. It's a complementary piece to it. So it recognizes the appropriate codons on the mRNA. So here's what a transfer RNA might look like. They always try to give them the googly pieces here, but really this is just a big one, super long RNA sequence. And so we call it a tRNA because it is an RNA that has the site for an amino acid to sit here. But we actually have the anticodon in a particular region that's going to match up with our mRNA sequence and help us out. If you really want to go with a tRNA and the enzyme there, like there's a couple of pieces kind of together that are working. It's just giving you a space-filling model. So translation occurs on the surface of a ribosome. We're bringing this back in as the ribosome is the site of making proteins. So ribosomes coordinate their functioning of mRNA and tRNA. So they help bring these together. And ultimately, the synthesis of polypeptides and then proteins. Ribosomes have two subunits, a small and a large. And each subunit is composed of ribosomal RNAs that we call rRNAs for short. Ribosomal subunits come together during translation and have binding sites for mRNA and tRNA. So let's take a look at what that looks like. There's a lovely space-filling model, right? But here's a little bit better. There's a small subunit and a large subunit that fit together. There are tRNA binding sites. There's an A site and a P site. And what will happen is that this mRNA strand gets fed through here. The tRNA will match up the codon with the green anticodon, and the polypeptide will be grown from here. So we can kind of work these two sites together to grow a polypeptide chain. Ribosomes of bacteria and eukaryotes are similar in function, and those of eukaryotes are slightly larger and different in composition. The differences are medically significant, so certain antibiotic drugs can inactivate bacterial ribosomes while leaving eukaryotic ribosomes unaffected. So drugs like tetracycline and streptomycin have been used to combat bacterial infections the same way. So an interesting connection there, little tidbit. So translation can be divided into the same three phases as transcription. Yay, a start, a get longer, and an end. So we'll go into a bit more detail because this one takes a little longer, which means that if you need to break, now is a good time for it. But initiation brings together mRNA, a tRNA, and the two subunits of a ribosome. Initiation establishes where translation will begin, and initiation occurs in two steps. The mRNA molecule binds the small ribosomal with a special initiator tRNA that binds with that start codon. So the start codon reads AUG, and the first anticodon, or the opposite codon, will be UAC. It's not as important. The biggest thing is that we've got the small subunit, the tRNA, and the start codon all coming together. It's in the second part that we actually get that large ribosomal subunit in, allowing the ribosome to function. The tRNA will occupy what's called a P site, or the polypeptide site. And then there's an A site, which is an available site or accepting site to receive the next amino acid bearing a tRNA. It's kind of a lot of words, but I'll show you what I'm talking about here. So here's the thing. We have some code here that's we don't really need, and the actual start of the message will be wherever we have AUG. Wherever that is tells us to start. I like to think about if we're doing a recipe, you know, if you've ever read recipes online, go look at one at some point. They'll always be like, this recipe is a great way to blah, blah, blah. I was like, we don't care, we don't care, we don't care. Start with 4 cups of, that's where we want to start. Start with however much, I don't care about, what's a great recipe for, just give me the recipe. So that's the actual start of the recipe is at AUG. Okay. So initiation, we have a start codon here. It's met with that tRNA that's got methionine on it, and we've got our small ribosomal subunit. Then we'll have a large ribosome subunit come in, and will set itself to where it can orient towards where this tRNA is in the P or the polypeptide site. Now elongation gets tricky because once this is complete, we've got to add on and make elongation happen. In order to save you some time, we can go through this. I would suggest that if you're really struggling, but let's see if we can do the shorthand, because I know we're practicing shorthand real quick. So each cycle of elongation has 3 steps. 1, anticodon of the incoming tRNA molecule carrying a tRNA pairs with the mRNA at the A site. So anticodon, incoming tRNA, pair mRNA at A site. Polypeptide will separate from the tRNA at the P site, attach new bond to amino acid carried by tRNA at the A site. So basically the polypeptide is going to move to the tRNA at the A site. Boom, polypeptide moves to tRNA at the A site. The P site tRNA now lacking amino acid leaves. So P site empty, leaves. The ribosome moves, and the remaining tRNA, which has a growing polypeptide from the A site to the P site. So basically what this means is, so here we've set everything up. Now we've got the next codon to come into the available or the accepting site, the A site. Once it's there and it matches up, the polypeptide that's here at the P site will move over to this one. So you see the little green arrow, it's trying to get it to move over to this new one. Once our new polypeptide has done that, this one will exit. So you can think the next thing is exiting. And we've got all this connected now at the A site. We'll then move it over, so that way this tRNA is now in the P site, or the polypeptide site, allowing the A available site to be available for the next amino acid. And it continues in that manner. So elongation continues until the termination stage of translation. Okay. Termination is when a ribosome reaches a stop codon. There you go. The completed polypeptide is freed, so polypeptide is freed from the last tRNA, and the ribosome splits back into separate subunits. So stop codon, polypeptide freed, subunit separate. Boom. So the flow of genetic information, good review. Notice this is review, so make sure that this makes sense to you and this is what you see. But the flow of genetic information is from DNA to RNA to protein. The transcription, DNA to RNA, the mRNA is synthesized on a DNA template. In eukaryotic cells, transcriptions occur in the nucleus, and the messenger RNA is processed before it travels to the cytoplasm. Prokaryotes, transcription would just happen where it is in the cytoplasm. Okay, hopefully we have this. It's a good time to update and check those notes. We can see the whole process, transcription. We have a start area. We build an mRNA strand, 5' to 3'. There you go. Then we have translation with anticodons, and we have the amino acid there. Okay, yeah. So we've got those together. Translation can be divided into 4 steps. Amino acid attachment is sort of what we looked at, but really initiation, elongation, and termination. Okay, so that's that sort of amino acid attachment. It's like an intermediate step. It's like a 0.5 step, whatever. Okay, then we start with things in the P site. We get the new tRNA that matches, that's anticodon, matches the codon on the pink mRNA. Then we move our polypeptide over to the new strand. Then it moves over until we reach a stop codon, and the polypeptide releases, and everything breaks apart and terminates. Ta-da! Last thing, let's talk about mutations real quick. So a mutation, this is actually really important to know. A mutation is any change in the nucleotide sequence of DNA. Mutations can involve large chromosomal regions, which we talked about in class a little bit, or just a single nucleotide pair, and it can have dramatic effects either way. A lot of words on this one. I'm just going to get to where I can show you pictures. Do I have pictures on this one? Yes. So you can use the word as a reference. I do want to shorten it up. So this is a single point mutation where you get sickle cell, is that glutamine in this one is changed by one single base pair, and we get valine in that same spot, and it gives us a sickled hemoglobin. Okay. These are frame shifts. Okay, I thought I had better ones for point ones. Okay, let me go back then. Okay, we have a couple of categories. So nucleotide substitution involve replacement of one nucleotide with its partner. Okay, so sometimes we can have one with no effect that we call a silent mutation. We'll talk about them a little later. The change in the amino acid coding is a missense mutation, produces a different amino acid. This was the sickle cell it just showed you. This is the missense where we actually changed the amino acid. We can have huge problems with that one. And then sometimes we have this nonsense mutation, and nonsense, I'm trying not to write underneath it there, it just produces a stop codon. So those are what we call, I lost it, point mutations. So all of these here are what are point mutations. It happens at one point. The other thing that we can have is when we insert or delete one or more nucleotides, you actually cause a frame shift, which alters the reading frame. And it can lead to some significant changes. So that's kind of in there. Mutagenesis we'll talk about a little bit later. And there's some examples here that you can take a look at where we've got a deletion and it shifts the entire frame over. Insertion, again, everything here is shifted. So let me end it at that, and we can discuss them a little bit later, but we'll end there. If you have any questions at all, because there is a lot happening, we'll try to review, and I'll give you some videos up online to look at. But if you have any questions at all or are not sure about what's going on, shoot me an email and let's figure that one out.